Hey everyone, it's Keith from GRIN. I mentioned earlier in the day that when new families get their diagnosis, they often go into the Facebook groups, the support groups, and find other families. And one of the first pieces of advice they get is that they should go and try to find out if they have a gain of function variant or a loss of function variant. And the two places that they will go to do that are either the lab of Dr. Stephen Traynelis in Atlanta at Emory, or the lab of Dr. Xavi Altafaj in Barcelona at IDIBEL. So we're very grateful to have them both here today to tell us about gain of function and loss of function, how they figure out what GRIN patients have, and what are the differences and what are the potential implications. So I think we're going to start with Dr. Traynelis, who will give a bit of an overview, and then we really want this panel to be dynamic. So there's that Q&A section up at the top right, and we're hoping to have lots of time here for questions. So we're going to hopefully start questions in 5 minutes or so. So get your questions in early, and we can make sure we get them answered. So Dr. Traynelis, over to you.
Great. Thank you, Keith. The first thing I want to say is that I appreciate an opportunity to participate in this meeting. It's just been terrific. The talks have been terrific, and it's been inspiring and really informative to hear from the various parents and patient groups. And so I'm very excited about this meeting. I've got the slides up. I apologize, they're a little bit smaller than maybe they should be, but I think I'll go through them fairly quickly, and then we'll get on to the Q&A session. The very first slide is really just to synchronize our thinking. You've already heard today from a number of the speakers about NMDA receptors and what they are. This is just to emphasize that they are the main mediators of communication between two neurons that are talking to each other. And we've heard in a number of sessions, there's multiple types, AMPA, kainate, NMDA. Of course, we're talking about NMDA in this session, and these are comprised of the GRIN1 and the GRIN2A, B, C, and D genes, which we've heard about. We know of about 700 positions in that gene with variants that are absent in the general population, but present in an individual with neurological condition. Let's see, it's a little slow on responding my moving the slides. Let's see if I can do it. So what we're going to talk about in terms of gain of function and loss of function is really what these receptors do for the cell and the body. And they're basically electrical signal generators. They listen to what one neuron has to say in the form of its released neurotransmitter, and they open up a path for ions to flow into a second neuron. And if we were just got a picture over here on the left, you can't see it very well, but there's a presynaptic terminal releasing glutamate onto a second neuron. And if we were to stick electrodes into the neuron that's talking and the neuron that's listening, what we would find is that there is a time course where current flows into the second neuron. And we can measure that. Xavi measures that. We measure that every day in the laboratory. And I've shown over here on the right on this slide a couple of plots. And this is just current flowing as a function of time. And you'll see sort of a blue highlight underneath a blue line. That would be the current that flows through an NMDA receptor, through a receptor made of the GRIN subunits, with every time one neuron talks to a second neuron, and we call that a synaptic current. And so when we think about gain of function and loss of function, we primarily think about the size of that blue area under the current. And a gain of function means that blue area somehow gets larger, it either gets higher or it stays around longer, or that blue area gets smaller. So I've got on this slide, I'm just trying to break that down to a real world fashion. Gain of function would mean a variation in a patient increases the amount of current that flows. I have a larger lightning bolt just to kind of think about electrical signaling. Loss of function would decrease the amount of current flowing, got a smaller lightning bolt. And if we just forgot about the receptors and we thought about cars, a gain of function might be anything that increases the speed of a car. So there's many ways to do that. You could press on the accelerator, you could lubricate the axle, you could shift the transmission into a higher gear, you could take off parts of the car, get rid of the doors to decrease the weight. Anything that makes it go faster would be a gain of function. And in the receptor, there's many ways to make it work better. Loss of function might be anything that decreases the speed. You could apply the brakes, You could let the air out of the tires so it doesn't roll very easy. You could restrict the gas flowing to the engine, or you could disconnect the muffler and let it drag on the ground and create friction. All of those things would slow the car down, but they all do something different to how that machine works. And so it's the same with these variants. Gain of function and loss of function is sort of a readout. It's a readout on what it does to signaling, but it can come about by many different ways. So Keith had set some questions up early on, maybe to get us started. I've got 5 or 6 questions. I'm just going to read through these, and then we'll take questions from the audience. So the first question, general question we had is, why is it important to know whether a patient has a gain of function or loss of function? Well, the answer to that is patient symptoms are really determined by what the mutation does to the protein, not so much where it is in the DNA. And so as we measure this, we're going to be able to group patients with similar changes together, and that will help us better understand the condition and better understand treatment. The second question, how do you determine what type individuals have? And what Xavi and I do is pretty much the same thing. We go into the lab, we make synthetic DNA in a test tube, harboring the change that's seen in the patient in the GRIN gene. We trick cells into taking that DNA up, and they will then manufacture the GRIN or NMDA receptors, and we can measure electrical properties of those receptors in the system with amplifiers and various equipment. Could treatments for all loss of function variants, for example, GRIN1, GRIN2A, GRIN2B loss of function, be more similar than treatments for variants in one gene, say GRIN1? And the answer is we don't know, but probably, because the driving effect of these GRIN variants is really what they do to protein function. And so it wouldn't really matter if the mutations were in the same gene so much as that they do the same thing to how the gene operates. Are there clear delineations between gain of function and loss of function? It's a little bit of a tricky answer because we can't measure everything the receptor does. Like a car, there's a lot of moving parts, and we can measure 7, 8, or 9 different features of the receptor, but we can't measure all 50 things the receptor does. So we pick the major ones. When we do that, about half of the receptors show a very clear, unambiguous gain of function or loss of function. 25% show no effect, meaning the variant may change something that we have not measured. And then 25 are mixed effects, where one aspect looks like it works better, but another aspect looks like it works a little worse, and we can use some math to make a guess, but some of those are more ambiguous. Keith also asked, how much do you think the human brain is wiring and trying to compensate? And the answer is that the brain works extremely hard to compensate for changes. And there is no doubt that some of the changes in circuit function that we would see in the patients would be the brain's attempt to correct for a defect in one of the receptors. And sometimes we call that a maladaptive effect. It tries to fix things, but it might make things worse than they were. 2 more questions from Keith. Is it possible that we might see different results from different laboratories? And the answer is, if the same test is run in 2 different places the same way, the same results will come out. And stated another way, in the 1980s and 90s, when these receptors were first being studied in detail, the properties were identical, whether it were done in Tokyo, London, Paris, Barcelona, Atlanta, wherever. If the same experiment's done, same results come out. And lastly, can what looks like a gain of function in a single receptor result in a loss of function overall in the brain? This is a really interesting question. And the answer is, in a way, yes. If a GRIN variant passes too much current, the brain may try to compensate, just as in a question ago. And that compensation could lead to an overall reduction in excitability. The neurons no longer talk to each other as efficiently, as well, or as frequently as they did. And so that gain of function variation in one of the GRIN genes could actually cause a suppression of excitability, depending on how the brain seeks to respond to that change. So that's the intro and sort of some initial questions. Hopefully that will both prime the pump for getting more questions, but also answer some obvious things right out of the gate.
Okay, thank you so much for that. And it's great to see so many questions coming in, and I'll get to those in just a second. But I wanted to ask Xavi, so Dr. Steve talked about what they do in his lab, how they measure the functional analysis. And do you use in Barcelona the same technique, or is it different?
Yes. Well, first of all, thank you for organizing this awesome meeting and all the organizers. So to address your question, indeed, we are using different cellular models in our lab. So Steve is using the xenopus side mostly, and he can do well. We're currently doing an amazing pharmacological study of the mutant greens, and in our lab we are using a complementary cell model, which is the mammalian cell model. But basically, the question is the same. So we try to express this mutation of green genes and to see how the NMDA receptors respond. And we can have well, we don't measure exactly the same variants, because in our cell model is a bit more complicated to perform pharmacology, but we have different tissues that we can evaluate in a mammalian cell system. And we also use primary neuronal cultures, and some other laboratories, they use different models. But at the end, the question is the same, is to characterize what is the consequence of a green mutation, and indeed the results are pretty similar.
Okay, thank you. So I want to get to some of these questions. So I'll just throw out the questions, and either of you can answer. But the first question is, what I guess some parents have had their kids' variants analyzed, and the results come back as inconclusive. So what does that mean, and what would the next step be for them?
I'll just take a shot and see what Xavi says. Inconclusive, often means that, for example, glutamate becomes more potent. And so you might think you're driving more current through the receptor, but then maybe in a mammalian system, you don't find as many receptors at the surface. And so you're left questioning, maybe there are fewer receptors, but they work better. And, the answer would be for that individual to, I guess it's probably not a very good answer, but you just need to wait until additional information comes out and, additional assays are done. There are other ways to look at that, but there's no way to parse that information when it comes out mixed. Xavi, do you want to add to that?
Yes, totally agree, Steve. So I think that one important key notion that in your slide, it was the net effect, and sometimes we can get some different aspects of the receptors. So it's like humans, we try to make some categories and to say, okay, this is a gain or this is a loss of function. But indeed, there are plenty of gray tones, and nothing is that absolute. So there is not like white and black, and we have mixed alterations. And this is why some notation, they can be considered as not conclusive, because for some aspects is well, they look like a loss of function, or some other aspects, they will indicate that it's a gain of function. So at the end, we try to make a much more complicated and to develop. So it's important to see that we have like this fast screening method to know if one variant is a very, like, let's say, pure gain of function or pure loss of function. But for those that are complex, we need to develop much more sophisticated models, such as using neuronal models or the mouse models, to know what really, what is the net effect in terms of neuronal physiology or behavior of the animals.
Okay, great. So we've got about a dozen questions in already, and some of these you'll be able to answer quite quickly, and some will be more detailed. So I'm just going to go through them from the earliest ones that came in. We've got a question: can a gain of function or loss of function be determined just through genome sequencing?
I would say no. You have to measure. You absolutely have to measure the function. You just cannot predict until you do the experiment.
The next question.
I would add that So I agree with Steve, but somehow we are quite lucky that during the last years there is a growing data on the functional annotation, especially from Atlanta. They delivered plenty of annotations of several mutations, and now for many families joining the GRIN community, they are writing Steve's lab or our lab or some other's lab, and they check for this information. They just provide the genetic variant, and going back to this database and the experiments that have been already performed, it accelerates a lot because we just for similar or the same variant, we can already say it was a loss of function. This variant, of course, for another patient that is requesting the analysis, the diagnosis will be the same.
Yeah, that's a very important point. Once it's done for one individual, that variant never needs to be tested again. Every individual that comes in with that variant could simply draw from that data.
Okay, the next question's in Spanish, but I think I understand it. So the question is whether whether people who have epilepsy or seizures and gain of function or loss of function, what's the connection or difference there? Did I get that right, Xavi?
Yes, you're completely correct.
Okay.
In Spanish. Well, this is something that Dr. Lemke especially is trying to do, to delineate and to make this association between loss of function variants as a variable, the different subunits, because it's not the same having a mutation on GRIN2A, that they have much more incidence of epilepsy than GRIN2B. But still, it's not that clear, the distinction between gain of function, a given gene, and the expression manifestation of epileptic seizures. I don't know if you want to complete the answer, Steve?
Just one comment. Often for GRIN2A, loss of function will drive epilepsy and hyperexcitability, and so will gain of function. And we're actually working on mice for both of those. And what we find is that there's a developmental change in a whole other cell population, the interneurons. And so the effects go to your question, Keith, about the brain compensating. It's seeing loss and trying to compensate in a way that drives excitation, and it sees gain, and the outright gain drives excitation.
Great. How accurate, someone asks, how accurate is the information provided after your testing? This person's daughter has a gain of function, but she says she's heard of other parents that have been misdiagnosed through the analysis. So their kids were diagnosed with gain of function, and after a couple of years, it turned out to be loss of function.
Yeah, I'd have to know exactly what cases those are. Once we make the electrical measurements, they're done. They're done in a highly rigorous fashion, and they're not going to change. What can happen is more information can come. For example, you can measure electrical current, show a gain of function, but not perform the surface evaluation. We can do rapid jumps on the mammalian cells, see that the signal is longer or shorter, but if you don't know whether it's going to the surface or going to the right place in the neuron, you can't be certain whether it's gain or loss, and maybe new information comes out later to shift that view.
Yeah, and one example I can talk about is just with my son Bryson, where the functional analysis was done at CFIRV, and he has a very complicated condition, and so CFIRV initially just put it as that it was unclear, which it was, because it seemed to be a gain of function with magnesium and a loss of function for glutamate and glycine, but eventually it was, I think even CFIRV looked more at it and saw some other variants in how they worked and suspected it was actually a loss of function. And then we also had a animal model that for this variant, a mouse, and brain slices done on that confirmed that it was a loss of function. So, so there are more, I guess, other ways to get answers as well, and an animal model sometimes could clarify with these inconclusive results. Okay, let me go back and see what's next. I think this one was already answered, but can a neurologist diagnosed gain of function or loss of function, I think basically the answer would be they can maybe help put you in touch with one of the labs that's doing the gain of function or loss of function, but it still needs to be through this functional analysis. Do you expect it will be easier to turn up loss of function problems or turn down gain of functions?
My view, and this is a scientific debate, very hard debate. My view is that for loss of function mutations, it can be pretty much easier to potentiate those hypofunctional receptors, because you just potentiate the overall content of receptors, and trying to reduce, and this is more for Steve, but to reduce the activity of gain of function receptors, you will also interfere with the wild type receptors, let's say. So my view is that, yes, I would say that reducing loss of function would be easier. Well, at least it's pretty difficult, but will be a bit easier than gain of function.
Mm-hmm.
What do you think, Steve?
Yeah. That's a good question, and there's not an easy answer. I agree with Xavi that on the surface it's easier to try to find ways to add to something that's missing. With pushing agonists, there are positive allosteric modulators in development in the pharmaceutical industry, gene therapies where you might be able to complement or drive up expression. So I think that's a reasonable view, but I guess I would also caution that although easy, it's hardly straightforward, and even the genetic approaches can drive up expression in the wrong place and create more problems than you might actually solve. So complex.
There's a couple of people who are asking, once they have the knowledge, once they learn that it's gain of function or loss of function, what do they do with that? How do they take it to their neurologist and figure out what to do next?
Well, a couple things, and then see what Xavi says. First is to share it with the neurologist and see whether or not there is a reason for the neurologist to alter the form of treatment that they're considering, or at least to view what they want to do in that context. The second thing they need to do with it is recognize its research data that is really laying the groundwork for how to solve these problems down the road. And so, much of this information is simply the beginning of understanding what's going on and what we can do, not necessarily actionable data. And so it's a difficult message. It's, it requires patience, but it's incredibly important, and there will be no progress without the information. So, share with their neurologist. At times there's something that can be actionable, and other times simply to know that, they're contributing. Stay tuned, come to meetings like this, follow what's happening, and be persistent as all the good parents are doing, and see whether opportunities arise as we learn more.
Yes. Okay. I think that's an important point here, because once they have the molecular diagnosis, they can go to the association. But I think that, at least for our experience in Europe, is that it's very important to create a network of clinicians that can be a reference clinicians for different states or countries, and they can give an advice to the specific neurologist that is somehow in a small town, and that doesn't necessarily knows what is a GRIN-related disorder. So I think that creating this relay between the scientists and the families by an expert clinician is very, very important.
We probably only have time for one or two more questions, but a couple of people are asking how to get in touch with the two of you to get these functional analyses done. And the first thing I'll mention is, as it has been mentioned already, that many of these have already been conducted. So it's possible that your child's variant has already been done and is available. So if you search for CFIRV, you can see the database there where Steve and his team have published all of their results. And I believe that Xavi is going to soon be publishing an open database where we can also go and see that. But if they go there and see their variants not there, what's the best way to get in touch with you guys?
You can get in.
It's twofold. One, there's a web. Go ahead, Xavi.
Oh, sorry. I think that yes, you can directly email the associations that they have the email addresses of the different centers, and this is the most easier way to contact us for molecular diagnosis or functional annotation. So something that is also important is that avoiding the redundancy. So if you contact one of the labs, it's good for us to know that the other one is already performing the experiments, because then it's not necessary to replicate the experiments, at least if they are like very, very clear. So yes, it's good if you can just say that, okay, Steve is already doing this and he found this experiment, so not necessary to replicate.
Right, and we now have to have our IRB requires approval through either UC Denver or Johannes Lemke, and so that is also an entry point. If you contact Tim and you sign up for that registry, then Jennifer will contact us and we'll immediately find out about it. And so there's two paths in for us to find out about it.
Well, thank you both so much. There's more questions. I know Steve is part of the Q&A at the end of the day, so maybe we can answer some of those there that we haven't gotten to yet. But thanks everyone for joining, and thanks to Dr. Altafaj and Dr. Traynelis.
Thank you very much.